Okay, let's fingers crossed for a few less technical swear words. I'm just going to send myself this link. In fact, no, I think I'm good to keep the chat on this screen. We'll see. What else can go wrong today? Previously, I've not been able to share my iPad, so I'm going to just do a little test on that right now. I'm just going to see whether it's going to work for me now that I've just tried switching over. But I'm currently using my regular setup, although my iPad screen is currently off, so that probably doesn't help. Just getting black old screen. Oh, I've got spinny wheel of death, which maybe means it's doing something. Doesn't look like it wants to play. I wonder, I've got one more possible option that might allow me to do this, but we'll see. When I tried this on my other computer, it also didn't like this as a method, but we'll see. Oh, it's now finding it. Okay, so there's probably, oh, but it's also giving me, that is super strange. So I wonder if it's the connection. Wow, interesting. Let me just have a look at this here cable. Looks pretty good to me. If this doesn't work, I will go through it the way that I did. I'll just screen share my computer, as in I won't be able to write the notes with you, but I'll be able to talk you through it and we can discuss them as we go. Let's just try this one more time. Okay, so we're gonna do some more at gene tech today, inserting target genes, maybe a bit on marker genes. It's weird, it detects the iPad here. Oh, but it just gives me a black screen, but clearly my screen is not black. I'm going to give up. I'm not going to spend all 20 minutes trying to figure that one out. Okay. I haven't seen any of the chat. Let me just catch up on that. My name is indeed Rich. Morning, everyone. I'm just going to switch to what is the quality drop in terms of when I switch to this camera? I've got two cameras mounted next to each other. I think this is going to be lower quality, but probably that's a great idea, Edith. So are you going to use the specification to create a checklist, basically? Okay, yeah, this is a 720p camera. The other one is a 4K camera. But I think if I choose the 4K camera, it might just have trouble sending that much data. I think the upload speed is a bit limiting. Okay, I think we'll go with that. So gene technology, let's summarize what we did yesterday. There's a couple of ways we can isolate a target gene. Why would we want to do it in the first place? We're going to want to do that because it's makes a useful protein. It might be for putting into a plant for commercial uses in terms of like a herbicide or a pesticide. You want that to produce that chemicals or that protein so that it's more resistant to herbicide or to pesticide. You can therefore get larger yield. Interestingly, we're talking about sort of the advantages and disadvantages or evaluating the use of something. You can use cost or money when you're talking about commercial processes, but basically not any other time. Obviously, you might also be inserting an enzyme that makes a certain thing, such as golden rice, to make, I think it was vitamin D or vitamin C that golden rice produces, that basically reduces like health disorders on diet. Lots of people in the world, I think about 60% of the world's calorific intake total comes from rice, just rice, 60% of the world's calories. So lots of people exist on a lot of rice, and if it's devoid of some nutrients, then it can mean that those populations are lacking certain vitamins, minerals. What else might we want to do it for? We might want to produce a useful protein for humans for medicine, insulin being a classic example. Vitamin A, thank you. Okay, we cut them out either using a restriction enzyme, which is going to find a restriction site, which is specific to its active site. It's a palindromic sequence, which means it reads the same forwards as it does backwards, and it will cut on a zigzag-like angle. It does not cut as a straight line like a judo chop meat cleaver. The purpose of that is to leave sticky ends, and sticky ends are these unpaired bases on the end, which obviously we've got sort of loose hydrogen bonds here, and it's going to find a complementary sticky end down here that's the same sequence, because if we use the same enzyme, we get the same sequence, and then they're going to get stuck together, and then we use an enzyme called DNA ligase to put it in there. Okay, so let's basically crack on with a bit of, I'm going to have to pull up. In fact, what I'm going to do is I'm going to go to the overview page in the content guide. I'm going to share that with you and sort of show you how to find the videos once inside the course. I'm also going to just pull out. Yeah, they are, restriction enzymes are specific to palindromic sites, but they're also, each restriction enzyme, there are many, is specific to a specific sequence of bases. Each palindromic site has its own restriction enzyme. Mum and dad is not a palindrome, Ruhab, because mum and dad begins M-U-M. Oh, like mum, okay, M-U-M on its own is M-U-M, yeah, and dad, D-A-D. They are individually palindromes, but the sentence or the phrase mum and dad is not a palindrome. I see what you're saying. So mum is a palindrome, dad is a palindrome, Hannah is a palindrome, never, odd, or even as a phrase is a palindrome, reads the same forwards as it does backwards. Okay, so if we have a look at the content guide overview page, I'll just sort of show you where inside you would find all these things. Let's just reduce this in size a bit. Race car. Okay, so we've got, these are obviously, I'm just in the AQA course because that's what I'm picking the content that is suitable to everybody though. This is gene technology as a section. We looked at isolating target genes yesterday. We're going to look at inserting those genes. So once we've chopped them out, we can either chop them out with that restriction enzyme, or we can use a reverse transcriptase to take the messenger RNA for that protein and convert it into complementary DNA for that specific, make it into complementary DNA for that gene. Okay, so these are my template notes. If you're one of the subscribers, you can download the notes template from underneath here. And obviously you can ask me questions if you do get stuck on any of this, because I'm going through it quite quickly. Chuck me questions in the course. For my subscribers, I will be resuming my regular live classes. I think I'm going to revert back to Thursdays. I'm going to make a decision on that today and send an email out to everybody. Okay, so if you're year 12, finished all the content, into the, I mean, this is deep into the A-level content. I mean, it's not going to hurt. If you're not bored out your senses, then I think following along, getting a first exposure now will be a good idea. I'm going to be going quite quickly, so it might be a little bit hard to follow if you're completely fresh and you're actually a year 12 student. But let's have a look at this, and I just want to check that. Okay, so inserting target genes, I'm just going to go over some of the basics here, then we'll discuss it, and I'll keep sort of screen sharing and then not screen sharing so you don't just like stare at the page the whole time. So inserting target genes, we've isolated them either using a gene machine. That's mostly an AQA only thing, so don't worry about that. The gene machine, you just like, you know the sequence of the gene that you want and you make it in a machine. A, T, G, G, C, G, and you just make the gene. Reverse transcriptase, this makes single-stranded complementary DNA, and restriction enzymes cuts out the double-stranded DNA. So we've basically got this. It's really important that when we cut out our gene, or if we're making our gene from scratch, for example, using a gene machine, then you include both the promoter region and the terminator region. This is the bit immediately before and immediately after the gene that, is that underneath my preview window? Can you see where my mouse is pointing right now? I don't know whether that's visible to you or not. I'm actually going to have to just stop sharing to figure that out because I can't see your answers. Okay, so I can use my trusty pen analogy. Now I'm going to, those of you who are just in my AS Live class, I use this way around. If the white bit is the gene, the white barrel of the pen here is the gene, then the bit immediately before and immediately after, I don't know if that's the way around, before and after for you, it's probably the wrong way around. So for you, this would be, if we're reading in this direction, the bit immediately before, this would be my promoter region. And then this would be my terminator region. Obviously, they look different here, but it's just a small sequence of bases immediately before and immediately after. What does the promoter region do? What happens at the promoter region when we're starting transcription? What's the significance of the promoter region? You could get this question asked here. You could get this question asked in PCR as well. Allows RNA polymerase to attach. Yeah, and when you say enzyme, Matilda, always, if you can, name the enzyme. So RNA polymerase is the one to say here. Okay, so now one of the things that we're doing there's two types of tests for SARS-CoV-19 for coronavirus. There is, you can test to see whether you have the virus inside you, or you can test for antibodies to see whether you previously has been infected, and then whether you have the antibodies for it, whether you've undergone a primary immune response for it. So if you take a sample of a throat swab from me and you're going to test for the presence of the virus, you're looking to see, is the virus in you in any decent quantity right now or not? What you do is you take a throat swab or a nose swab, get those basically sample of tissue from the mucous membranes, and then you're going to put it through PCR. You're going to have the viral genome is RNA. So you're going to be basically doing PCR, but it's going to be doing it RNA rather than doing it on DNA, which is what we learn in the spec. There's going to be loads of DNA in there. There's going to be all of my DNA in that swab. There's going to be all the bacterial DNA from all the bacteria that live in my throat and my nose, and all those types of things are going to be in there. How do we only make more copies of the viral DNA? We want to amplify, we want to make more copies of the viral DNA. You could use a DNA, well, you'd have to use an RNA probe technically because we're detecting for RNA, but you could use an RNA probe. But really, before you do that, you might only have a few bits of virus particle in there, a few bits of virus RNA. You want to make lots of copies using PCR. Yeah, exactly. And how would you ensure that you only make more copies of the viral RNA rather than making copies of everything that's in there, all of my DNA, all the bacterial DNA? You would only include the primers which are the complementary to the promoter regions of the viral RNA. So when only the primers for the viral RNA exists, they're going to be the bits that form little double-stranded sections at the beginning of the gene or the RNA sequence during the beginning of PCR. And that means that's the only place where the RNA polymerase will be able to bind on and then create more. In fact, it wouldn't even be RNA polymerase, it'd be a different enzyme, the equivalent of DNA polymerase in RNA. I mean, we don't need to know the specifics of how you would do it to RNA, but in terms of translating what we know about PCR, you'd only make more copies of the viral DNA because you'd only include the primers complementary to the start of the viral bit that you want to copy. Primers are the bits that are complementary to the promoter region. So when we're doing PCR, first step, let's do it with DNA, it's way easier. We've got double-stranded DNA, you heat it, to make it single-stranded. Now we've got 2 single-stranded bits of DNA. Now we've got the gene or the bit of DNA that we're interested in copying. And immediately before that, we've got our promoter region, which is where the RNA polymerase would normally bind on to do transcription. But the primer is the bit that's complementary to the promoter region, which will add here just by the hydrogen bonds. We'll bind to each other and they'll stick on. And now we've got a little bit that's double-stranded. The promoter region is now double-stranded. You've got the promoter and you've got the primer below. You with me so far? Yeah, so the primers are DNA pieces, single-stranded, that are complementary to the promoter region, which is before the bit of DNA that you want. This is a totally free class. And then you've got our double-stranded. Now the enzyme can bind on. The DNA polymerase can bind on where it's double-stranded, and then it can start reading along. So if you've got a whole mixture of DNA in your starting pot for PCR, human DNA, bacterial DNA, you've got all sorts of stuff floating around in there. If you only include the primers for the bit that you're interested in copying, none of the others will copy, because none of the others will end up with this little double-stranded section. The DNA polymerase won't be able to attach anywhere else. So that's how you can choose which bits to amplify, by just including the primers for the bits that you're interested in copying. If you cut out the DNA fragment using restriction enzymes, do you have to add a promoter and terminator? No, because if it's in DNA itself, it's going to include that gene will have immediately before it. There won't be a restriction site between the beginning of the gene and the promoter. They are basically butted up against each other with nothing in between. Whereas you then, the restriction sites are going to be further past the terminator or before the promoter. Yeah, you would in the lab, they would probably have sequenced the entire length of DNA they're interested in. So they would basically make the primers in advance. Yeah. Fluorescence. I'm running out of mustache wax. It's terrible. I don't know what I'm going to do. Look at it. It's like this just doing its own thing. Like, this one's all frayed. Least of my problems right now. I need a beeswax candle. I usually make my own mustache wax, but I need some beeswax, and I've just arrived back in the UK, so it's not exactly a high priority. Yeah, if you were to make the gene with a gene machine, you would need to make sure you included the promoter region and the terminator region to tell the DNA polymerase where to start and where to stop, or where to tell, yeah, the RNA polymerase where to start and where to stop if you're making the gene. Okay, well, it depends. The primers are only involved in PCR. The promoter region and the terminator region are involved in protein synthesis. Does that kind of clear things up a little bit? It's a little bit, this is where your knowledge and understanding of those key terms is the difference between primer and promoter, or the difference between DNA polymerase and RNA polymerase, and where things are taking place. It's like, it is important that you understand them at the very least. Okay, so gel electrophoresis. Okay, so often you will use PCR in conjunction with electrophoresis, but that's going to be for DNA profiling or DNA fingerprinting. In fact, that's going to be you testing the length of how many variable number tandem repeats we have at a certain non-coding section between the genes. If go back to the live class I just did for year 12s, the recording will be available soon. That links back into that quite nicely. You do need a promoter region in PCR because it's the primer that is complementary to the promoter region, which tells the DNA polymerase where it can bind on to start copying the extra copy for PCR. Edith, come back to me about gel electrophoresis. Let's not get too sidetracked for right now. I want to try and get through the rest of sort of how do you get the target gene that we've chopped out into a vector. So a vector, wherever you come across the word vector, this could be in terms of disease transmission, a mosquito, Aedes aegypti, that is going to transport that disease from one to another. It's a transportation mechanism. In gene technology, a vector is something that transports our target gene from one place into another place, usually from one organism into another organism. So that could be, most commonly, it's a plasmid. 90% of the questions you're going to get that include vectors will be about a plasmid, obviously double-stranded loop of DNA, usually found in prokaryotes. Can anyone give me any other kind of vectors? A virus. So a bacteriophage is a virus which infects a bacterium. So yes, we can totally use a bacteriophage or a virus. You probably wouldn't need to be as specific as to say a bacteriophage, but we could say bacteriophage or virus. There are also some liposomes, I think, that you can use. OCR, there was one question from OCR in the old spec that was just the most insanely detailed question on, I didn't know most of the answers, and I don't know, I think it was so badly answered, I don't think they'll ask it quite in that much detail again. Okay, so the vector is the thing that's going to move the DNA into the bacteria. So a lot of you saying bacteria. The bacteria will be the host organism, transformed organism that will produce our gene, our proteins for us. We're going to basically use the bacteria to convert our gene into protein. We're going to use the bacterial cellular machinery to make the protein. But the thing that moves the gene, it's being moved into the bacteria. So the bacteria itself is not the vector. The vector is the plasmid or the bacteriophage virus that does that. Yeah, a mosquito is a vector, but it's a disease vector. That's moving malaria from one place to another. So in this case, in the context of gene technology, you couldn't say that a mosquito is being used as a vector for gene tech, but a vector is universally used in biology for something that moves something from place A to place B. Just depends what you're moving. Birds can move disease as well. Yeah, absolutely. Bats. Generally, things that can fly are going to be the most spreadable. Yeah, I'd say a vector moves a gene into a host organism's DNA. What do we call DNA that's a mixture of two different, I'm not going to say species, I'm going to say organisms. Yeah, I can't share my iPad today. I will show the notes so you can take a screenshot, but we're going to do it in more discussion format today. Just going to try this one instead. Recombinant DNA. It's a recombination of DNA that didn't exist before. And when we put that DNA into an organism, what do we call the genome of that organism, or that organism as a whole? We call that a transformed organism. Its genome has been transformed. So typically you're going to, in the newspapers, you'll see genetically modified. Really, you should be saying it's a transformed organism rather than a genetically modified organism. Some mark schemes will accept genetically modified, but they're looking for transformed on the whole, or transgenic, either of those. That would be accepted. Okay. Does the plasmid need to be resistant to antibiotics? No, but you can use antibiotics to detect whether this works. So basically, we get our target gene, we've chopped it out, isolated our target gene. We're then going to insert our target gene into the vector. For the sake of argument, I'm going to say we're going to use a plasmid because that's where most of the questions are. So we're going to insert our target gene. The easiest way and the most common way they're going to ask the questions on this is that we're going to use a restriction enzyme to cut out our target gene from the host DNA. What do we get when we use a restriction enzyme that we don't get when we use reverse transcriptase or a G machine? Sticky ends. So we've got these unpaired bases. On the top, we've got one over here, and on the bottom, we've got them over here. It'd be easier for me to do it like this. We've got our unpaired bases like this. What could we then do if we want to insert that into our plasmid? Plasmid also double-stranded. Unpaired bases, sticky ends. Exactly. That's what we've got. What do we need to do in order to chop out? This would be so much more convenient if I could draw it, but I can't right now. So We've got to use exactly. So we've got our plasmid, we've got a ring, circular ring of DNA. And if we use the same restriction enzyme, then it's going to be complementary to the same restriction site, which will mean it will leave the same sticky end. So we're basically going to chop it open like this, and we're going to have a gap, and we're going to have sticky ends on our plasmid as well. And those sticky ends are going to be complementary to the sticky ends from our target gene that we cut out from before. And that means my unpaired bases here are going to match my unpaired bases here, and then they're going to basically stick to each other with hydrogen bonds. But the thing that won't be connected will be the sugar phosphate backbone. Yeah. So we need to seal up. We need to sync up that sugar phosphate backbone to make it a continuous loop of DNA. So what enzyme do we use to reseal two lengths of DNA? We're not adding on a single base. We've got a long chain of DNA, another long chain of DNA. We want to link those two together. There's DNA ligase. Yeah, exactly. So that's the difference between DNA ligase and DNA polymerase, is that DNA polymerase is going to add one base on, not connect two long strands. Yeah, Rehab, go check out the content guide. I will do as much as I can. I'll share the notes, but I can't. My iPad is just giving me a black screen when I share it on either. I've got my new laptop, I've got my old laptop, and nothing is happening for me. So I've got a pen as a prop, and I've got my voice to describe things, and my hands. Well, obviously, if you're really keen to catch up on all the content, then the subscriptions are still there for you to use. The whole course is ready to be taught to you by me with full color notes in the course. But yeah, for the rest of you, I'm here on YouTube. I think we're going to do some changes to the way we do things on YouTube a little bit. We'll complete this week. Yeah, I don't know. Everything's kind of in flux right now. I want to do some more for my subscribers inside the course because I've been a little bit neglectful of those. Yeah, I will watch this space. We'll maybe do some other things on YouTube, maybe some more quizzy type stuff in the evenings, maybe get some parent challenges. But then when I finish this class, I'll ask you about some ideas that we've had, maybe. Okay, so the palindromic sequence is the sequence on the double-stranded DNA, because it's got to be double-stranded because it's the same forwards as it is backwards. And then when it gets cut out, they are the palindromic sequence. Half of the palindromic sequence is the sticky end. Okay, what next? So we've got our recombinant plasmid. We've inserted our target gene. We've used the same restriction enzymes. The complementary sticky ends were sealed together using DNA ligase. How do we get that plasmid into a bacterium? If I just came up to you and say, would you like this mystery gene? You don't know what it does. It makes a protein. In fact, it's going to make many proteins, but it's going to make the protein that we're interested in. Would you take it? Would you just take a random gene? Might be good for you. Might not be very good for you. You're probably going to be pretty reluctant to just go lucky dip on a random new set of genes. Might be really good for you, might be really bad for you. So generally speaking, bacteria are pretty reluctant to take in random plasmids that there's just given to them off the street. So what they do, basically, you need to stress them out a little bit, is more or less what you need to do. You need to stress them so that they're like, oh shit, I might die here. If I put a gun to your head, do you want these random genes? They might make this gun go away. You'd be like, yep, okay, I'll have it. You could welcome to take a few more risks when you're under stress. So typically use ice cold calcium chloride and then a heat shock. So you go from very cold in a calcium ion calcium chloride environment, and then you heat them up quite quickly, and then they just go into panic stations, okay, and they just start taking anything because it might be the solution to my problem. So it's a pretty minor point, the transmission details, but they are definitely, it does pop up from time to time. How would you increase the chances of getting this plasmid inside? And even then, even with the ice cold calcium chloride bath and then a heat shock, the percentage of bacteria that take up the plasmid and successfully become the transformed bacteria is still very low. way less than 1%. Like 99 out of 100 are still going to be not taken up the plasmid. They're just going to be the regular old bacteria. So how do you tell? How do you go about telling which of the bacteria have become transformed? To look at them down a microscope, you're not going to know. Use of restriction enzymes, isolate genes, leaves DNA with unpaired bases, these sticky ends. Oh, very nice, you're explaining. You use a marker gene, and there's two common marker genes that you're going to come across: UV fluorescence and an antibiotic resistance gene. Either or. So you're not only, so this is an additional gene that we add in addition to the target gene. We want it to make, let's say, the insulin. That's our target gene. But as well as the insulin gene, we're going to insert this marker gene, which could be UV fluorescence, or it could be antibiotic resistance. Let's use antibiotic resistance as an example. So now we've inserted both of those genes. Only the bacteria that have taken up the plasmid, the recombinant plasmid, and become a transformed bacteria or bacterium will contain this plasmid. So now, if we add that antibiotic, let's say it's an antibiotic resistance to penicillin. Obviously, lots of things are resistant to penicillin, but let's say it's penicillin resistance. You've got your culture of billions of bacteria on your plate. You pour on some antibiotic, some penicillin, or actually what you'd do is you'd put the penicillin in the agar that you were culturing them on. But either way, the concept is exactly the same. Only those that are resistant to penicillin will survive. So only those that have the penicillin resistance will survive. And if they've got the penicillin resistance, then they must also have the gene for insulin. So that it's a way of identifying. It's a gene that you can spot easily. UV fluorescence would be the same. You would grow your bacterial lawn of transformed and non-transformed bacteria together immediately after you've tried to introduce your target gene, shine a UV light on it, and then that 1% that did take up the plasmid will start glowing or fluorescing, and then you can culture those ones. You can pick them up and then use culture the glowing ones, because they have the glowing gene. They also therefore have the target gene, and then when you produce them en masse, they will produce the protein for you. Shall I go over the notes for you? I mean, I want... Interesting. I'm going to have to find them again because I seem to have... What is going on? Oh, okay, it's over here. That's why. Okay. So I can't see the chat whilst I'm showing you this full screen. We've talked about isolating them yesterday, inserting them. Let's just actually, I'm just going to skip to the end. Get my ugly hand out of the way. Inserting them. A vector is something that is used to move DNA from one place to another. We can use a virus such as a bacteriophage. So a bacteriophage, phage means eat. It's a virus that eats bacteria. So this is actually a virus. Plasmids, these are double-stranded loops of DNA that transfer genes between bacteria. This is my plasmid drawn over here. You use the same restriction enzyme to cut out the plasmid. That means the sticky ends will be complementary. So the same one that you used, the same restriction enzyme that you used to isolate the gene in the first place, remembering that we need to include the promoter region and the terminator region and sticky ends, as well as a marker gene for this to be successful. So if you used a gene machine, then we need to make sure that we've included those things in our plan when we were designing our piece of DNA to be produced by the machine. We then use an enzyme called DNA ligase to reform the phosphodiester bonds. Always a mark for the DNA's ligase, always a mark for saying phosphodiester bond, and always a mark for saying recombinant DNA. So this was our DNA to begin with up here. We have our target gene in green, we have our promoter region and our terminator region in blue. Then there's a black is just sort of normal DNA, and the red is my palindromic recognition site. So our restriction enzyme is going to cut here and cut here. This needs to be the same restriction site. This would be the same enzyme. If you use a different enzyme here, we'd actually need to make 2 cuts in our plasmid, but I'm not going to go into that much detail right now. So we've got our target gene. Really, I think we've got our promoter region and our terminator region, and then we've got our unpaired bases on the end, our sticky ends. We're then going to use the same restriction enzyme to cut the plasmid, and obviously we make the same cut because it's the same enzyme. This creates a flap, and then we insert our target gene with its. This one has got the unpaired base on the top here, and you'll see that when we do this cut, this one will be missing its top section. So this bit here is going to partner with this bit here, and then our bottom sticky end here is going to partner with our top sticky end here, and then we use DNA ligase to join the phosphodiester bonds to make a recombinant plasmid with our target gene. We'd also include our marker gene in there as well, our sticky ends, which are now just been merged into our plasmid. And then to get our plasmid into our bacteria to form our transgenic bacterium, then we're generally going to use ice cold calcium chloride and then heat shock. So make it really cold, stick it in some calcium chloride, and then take it from there, and you heat it up pretty quickly. And this basically increases the permeability of the bacterial cell wall to the plasmid. Five, four. I don't expect you to complete those notes in the time that I've taken. You just have to come back to the recording. You can pause it there. And the red bit in the ring is sticky ends. Yep. All the bits that were sticky ends before they were repaired up again. Okay, I can go through the marker gene set video as well. Let's do that. Why? Okay, let's actually, I can just. Okay, so this is the gene technology section of the AQA course. Obviously, your videos will be in a different order, and obviously sometimes different videos, sometimes very similar, but very different videos in terms of the level of detail. The next lesson in the gene tech section. Obviously, if you want to come and study these later on, this is the course. You've got PCR, and so someone who was asking me, was it Edith asking me about gel electrophoresis? Gel electrophoresis is actually covered in the apparatus and technique section, because that's where it's actually covered in the spec, is electrophoresis in here. Okay, let's take any questions there. I don't understand what your question, Rehab. Are those sticky ends of a different bit? Can you phrase it into a question that I can grasp in one go? Yeah, so the plasmid is cut open with the restriction enzyme, the same restriction enzyme, and then the target gene is cut out using restriction enzyme. And if it's the same enzyme, then it's the same active site, and therefore the top overlap of the sticky end is the same as the bottom overlap, or complementary to the bottom overlap of the other bit that was cut out, and therefore you'll get hydrogen bonding because there'll be complementary base pairing between them. Edith, I'm probably going to reorganize the OCR section to include an apparatus and techniques section, in which case it will be in the gene technology section for you. I can pull up the OCR course and show you at the end if you still can't find it. But let me go through this marker genes, and then then that will take us to the end of this session. I can then, or I can go through this, or I can do a question with you. I won't be able to write down stuff. I can share. I'll let you choose on that, whether you want. Yeah, those of you that don't have the course, I suppose this is a little sort of virtual tour inside. There's sections here, all the stats skills, the math skills, apparatus and techniques we've got here, all the AS sections. and every video looks like this essentially. This is 6 minutes 17 seconds. It's pretty typical, 6 to 8 minutes for a video. The note template for the video, the specification underneath, and then obviously the option to ask me questions on any of the content below, and then yeah, all the AS and all the A-level stuff. And then if I write you a video reply, then you can get video comment replies. So like when I can't explain it to you in a text, I'll just record you a little screen recording and send it to you down there. Well, let me stop screen sharing then. I'll see if I can find the marker genes question by OCR. Where's my... And I will endeavor to get my iPad working on my new laptop tomorrow. Let's open up the 3 of these. Oh, shift, not control. There's an Edexcel question on this that I can share quite easily. Okay, let me just share this. It's on my screen because I pulled it up previously. Okay, so we start this question with a flow diagram, I think. Oh no, I shared this one with you yesterday anyway. So let me stop sharing that, and I'll find. Question here. Any other? I'll just share this with you as I go. Okay, this is an OCR paper, new spec, A-level paper 3. Scientists have genetically, let's make it a touch bigger. Genetically engineered many different species of plant. Aubergine plants, Solana melangina, can suffer damage from moth larvae. Scientists have produced a variety of aubergines that are resistant to moth larvae. So they're resistant to moth larvae. To create the resistance, scientists have transferred a gene from Bacillus something or other, from a bacterium. Describe the process scientists could use to produce the pest-resistant aubergine. So how many marks was this? 6 marks. So this is basically describe the process that you can remove a gene from one organism and insert it into the other organism. The thing that we need to be careful of here is that we don't just describe inserting it into a bacterium. Here we've taken the gene from a bacterium and put it into a plant, but many of the same principles are going to apply. This, if you are an OCR student, is a quality written communication question. I don't worry too much about that. Your level of content, the detail that you include in the answer is going to determine your level. So level 1, 2, or 3. And then you're going to get the upper or lower mark within that level. So 5 or 6 is level 3, 3 and 4 is level 2, depending on how well you've written it. So most important is the content. And then second most important is how you've written it. They're the level descriptors. So indicative scientific points. So it's not marked on a point-by-point basis because it's marked by level. Only AQA students won't understand that very well. It's the same as the essay to some extent, but it's obviously a mini version. Method for gene extraction. Conversion of messenger RNA into complementary DNA with reverse transcriptase, or removal of a gene using restriction enzymes. One of the two. You don't have to give both. Use of an appropriate vector. A TI plasmid, I don't know exactly what that is. I might possibly do a bit of research into that. Or you can use agrobacteria tumofaciens. This is a specific plant vector for inserting into plants. Electroportation. Okay, so that's again why they've included that and not included. This is electroportation is another way to get a plasmid inside of a cell. So that would be the equivalent to the ice cold calcium chloride and heat shock. These are indicative points. They are not, if you've written use ice cold calcium chloride and heat shock, I think that would still be creditable. That's typically used for bacteria. Electroportation is an alternative way for inserting something into a cell. It doesn't actually have to just be a vector, a plasmid. You can use, OCR students might be familiar with electrofusion from animal cloning, similar process to electrofusion. This says TI plasmid. If someone wants to Google that and tell me what the TI is, it rings a bell, but I can't recall it. Tumor inducing, thank you. DNA ligase, a reference to marker gene and their purpose. Again, so this is pretty vague. And then electrofusion. Okay, so I'm just going to Google these. Electroportation must be the, what are they using? Electro. You can tell how uncommon this is, portation, because I'm Googling it. Microbiology technique in which electric field is applied to cells in order to increase the permeability of the cell membrane. Okay, so electrofusion. So I was right with that. I have to put biology in there. The fusion of plant protoplast by electrical stimulation. As you've got a levels response answer here, then you basically can answer. If you answer with correct science in a what's the word that they use? They use OCR use. It's not holistic. That's the AQA buzzword for the essays. Yeah, basically, if it's well-rounded and to A-level spec detail, then you'll be getting high-level responses. 6 marks. AQA will not ask you about electrofusion or electroportation. It's not. Okay, let me find one more question. This is pretty classic from what I remember. Okay, so silkworms secrete fibers, which are harvested to use to manufacture silk fabric. Scientists have produced genetically modified silkworms that contain a gene from a spider. So they're taking a gene from a spider, they're putting it into a silkworm. GM silkworms secrete fibers made from spider web protein, spider silk, which is stronger than normal silk fiber. So basically the spider gene is going to make spider protein. The spider protein is stronger than the silk protein. The method is shown below. Isolate gene from spider. It doesn't go into any gene details, but we're just taking the gene out. Could be any one of the methods that we've described. Insert the spider gene into a plasmid with a marker gene called EGFP. This is fluorescent protein. What does the G stand for? I can't remember. Green fluorescent protein. And it glows brightly under UV light. Inject copies into the plasmid. Inject copies of this plasmid into many eggs of the silkworm. Allow silkworm eggs that have taken up the plasmid to grow and produce spider silk. Okay, so I'll only let you see the bottom bit there. Suggest why the plasmids were injected into the eggs of the silkworm rather than into the silkworms themselves. if I inject you in the, okay, so we're making silk here. You might not know that much about the anatomy of silkworms, but you can imagine, just picture a spider making a web, right? Where does the silk come from? Silk is going to come from the silk glands, which are basically right on the arse somewhere. Yeah, it doesn't come out of either legs, doesn't come out of the abdomen. So where are the genes that are expressing spider silk? Where do we want them to be turned on? Yeah, so if I inject you in the foot with, I don't know, genes for rod and cone cells, there's not really any use for those genes to be there. So the genes have to be in the cells and turned on where you want them to be expressed. In this case, if you inject a silkworm with these recombinant plasmids, we don't know that they're going to get to be in the cells that they want. So why, if you put them in the egg, what's the benefit of them being in the egg? Protected, yeah, but what does an egg do? It's going to divide, and it's going to divide, and it's going to divide, and the genes that were in those starting cells are going to be copied by mitosis and copied by mitosis, and so they're in all of the cells, or certainly the vast majority of the cells. So you can assume that if the gene is in the egg, then when the larvae hatch, all of the larval cells will have that gene. So you're basically you're putting it in at the beginning so that those genes will be present in the cells where you want them to be present. So the answer on the mark scheme, gene gets into almost all or all of the cells look at how badly the mark schemes are written. So gets into cells that make silk. So that the silk producing cells will contain the genes. I mean, that's the ultimate point of it. They will all have the genes, but remember, genes will only be switched on in some cells. All genes are not all on at the same times. Suggest why the scientists use the marker gene. So why do you use a marker gene? Classic question, 2 marks. I don't have much time. So not all the eggs will successfully take up the plasmid. Silkworms that have taken up the gene will glow or will be identified. The scientists ensured the spider region was expressed only in the silk glands. What would the scientists have inserted into the plasmid along with the gene? What do you add as well as the gene to ensure that the spider gene is only expressed in the silk glands of the silkworms? I don't really like this question, to be honest with you. Okay, so you've got 2 options. Something you'd insert along with the spider gene. You've got the marker gene, although we've just answered a question on the marker gene. Okay, so to ensure that it was expressed only in the cells with the silk glands. Does this theory quite stack up to me? Well, the target gene is the spider gene. The marker gene, we've already answered this question. What else goes in with the target gene that's not technically part of the gene? And without it, the gene will not be expressed. It's not an epigenetics question. It's not primers, you're close, but it's not the primer. What's the primer complementary to? The promoter region, because if the promoter region isn't there, then the RNA polymerase will not be able to bind to it. But why that ensures that it is only expressed within certain cells I don't feel like this sentence is useful to us at all. Okay, cool. Right, I am going to let Ronnie have the room. And I'll see you tomorrow. Oh, I don't need to end the live like that, otherwise it won't actually end the live. I need to go to the control room. Try and find the control room. Live streaming here. Voila. See ya.